Thank you for the introduction. It's really nice to be at a symposium to hear about so many different fascinating examples of plant symbiosis with other organisms. Now, before I tell you about the particular system that I study, which is the nitrogen fixing symbiosis in legumes, I just want to step back to see that symbiotic interactions are widespread in nature. They are the rule rather than the exception. Higher organisms like animals and plants very commonly engage beneficial microbes for nutrient acquisition. Okay? So on the right, we can see that we as humans employ a large number of bacteria in our gut to help us break down the food that we ingest for better absorption. Okay. On the left, the plant also engages a multitude of soil microbes for its own nutritional needs. In this case, mostly minerals and ions, as well as water from the soil, which the microbes will help the plant absorb, which then drive photosynthesis. Okay. So of all those nutrients that are required for the plant to prosper, one of the most important and most limiting one often is the element of nitrogen. Okay. And this need is solved in the legume-rhizobia symbiosis by their interaction, which is one of the most successful mutualisms that we know in nature. So the two partners, the plant and the bacteria, can both be free-living organisms. But when they come together, okay, the plant then develops this dedicated symbiotic organ called nodule, usually on the root. Okay. And bacteria then enter this nodule where they find shelter as well as fixed carbon from the host. And in return, they convert nitrogen into ammonia for the host to consume. So this symbiosis developed many million years ago, and nowadays it's become a very elaborate developmental process. And that is because the bacteria in the symbiosis are acquired horizontally. That means that every plant, every legume plant, in order to form this symbiosis, will have to go survey the soil microbiome to find the right bacterium that is compatible with it. Okay. This is usually ensured by an exchange of chemical signals, for example, the nod factor produced by bacteria. Once a plant perceives this correct partner in its neighborhood, then this root hair will then curl to trap that bacterium. And the membrane will evaginate to form a tunnel called infection thread, which extends through the length of the cell and pass it toward the interior of the plant root. Okay? And it is in this infection thread that the bacteria will multiply, proliferate, and travel, if you like, and across cell layers. At the same time, the root cell in the cortex will now become a group of stem cells. They will proliferate to form a small bump, which is the incipient nodule. Okay, it is these cells, those new cells, that are destination for those bacteria traveling inside the infection thread. Once they reach those cells, the bacteria will be released from the infection thread to enter the host cytoplasm. And that's where they begin their nitrogen fixing process. Okay? So inside each host cell are hundreds of those individual bacteria. They are surrounded by a host-derived membrane and then turned into essentially a nitrogen fixing organelle that we call the symbiosome. Okay, so this is a very complex process, okay, but it did not just evolve all of its own. So we now know that it is related to a much more ancient and much more widespread symbiosis with soil fungi called arbuscular mycorrhizal symbiosis. So most land plants engage with this with those arbuscular mycorrhizal fungi. And it's much simpler than the root nodule symbiosis, in that in the fungal symbiosis, no new dedicated organ is required. It only just happens in the root, and it requires no new cell as well, either. Okay, so the fungus simply penetrates through several cell layers and reaches, again, the cortex. And in this cortex, that the fungal hyphae will bifurcate and eventually form a tree-like structure that we call arbuscule. And also, the fungus does not cross the host membrane, so it only stays on one side of the membrane. Okay, we now know that this fungal symbiosis was established around the time that plants started to colonize land, and probably helped the plant to achieve that. And it is later, much later, between 50 and 100 million years ago, that a particular lineage within the eudicot developed this ability to further engage bacteria to form nitrogen-fixing symbiosis. Nowadays, it's mostly legumes. Okay. So if we put the two symbiosis in its evolutionary context, we can see that legumes simply co-opted the ancient machinery used for arbuscular mycorrhizal fungi. It kept the pieces that it needs, okay, and then developed new functions that to engage with nitrogen-fixing bacteria from this second symbiosis. Okay. Another question becomes to understand which the functions between the two symbioses are conserved, and which are the ones that the legume developed later for this novel legume-rhizobia symbiosis. Okay. To start with, we now know that the initial recognition between the plant and microbes are very similar, whether we're talking about bacteria or fungus. So the bacteria produces nod factor, and the mycorrhizal fungi produces mic factor. Those two signals are structurally very similar. On the host membrane, they are perceived by very related, very similar proteins. Those are receptor-like kinases with lyse M domains that bind those symbiotic factors. Following the initial recognition, then the signal converge into the common pathway that involves a second receptor, ion, mostly calcium oscillation, a kinase activated by the calcium, and downstream transcription factors. So here, the signaling pathway diverges, but through related but distinct transcription factors for rhizobia symbiosis and mycorrhizal symbiosis, respectively. Once the plant perceives the presence of beneficial microbes, either fungi or bacteria, it then initiates a program to accommodate the partner. Okay. In the fungal symbiosis, you can see on the left, the plant also makes evagination, a tube-like structure to accommodate this filamentous fungus. Okay. On the right, the bacteria are not filamentous, but nonetheless form a file, a very narrow file, except at the end of the tip of this file, the bacteria are released and cross host membrane and are surrounded by this symbiosis membrane. In both structures, we now know that a number of proteins from the host are required. So, for example, this 3-protein complex made of a viperin that interacts with actin, a E3 ligase called lin, and exocyst component called exo70, they are required for both structures to maintain and function. Okay, so we can call them homologous structures. Of course, macroscopically, what's different in the nitrogen-fixing symbiosis is that the legume produces a delicate symbiotic organ called the root nodule. Okay, so it's only recently that we know we got some idea about how this new structure, new organ is constructed. It turns out that the legumes repurposed genes required involved in root and lateral root development to build this nodule. For example, in this case, you can see that a pair of transcription factors called short roots and scarecrow, they are usually, in all species, they are required to specify new self-lineage at the tip, the root. But in legumes, these two proteins, two genes, can be reactivated after the plant perceives the rhizobial signal. So they are induced both transcriptionally and post-transcriptionally. They form a complex, and then they drive cell division, which of course then builds the nodule. So you might ask that why does the bacterial symbiosis requires a specialized organ, whereas the arbuscular mycorrhizal symbiosis does not? Okay. It may have something to do with the quirks of the bacterial enzyme that does the nitrogen fixation. So this nitrogenous enzyme is a complex that consumes a lot of ATP as well as reducing power to split the dinitrogen gas molecule in the gas in the air and convert that into ammonia. Okay. But this enzyme is very sensitive to the oxygen that's also in the atmosphere. So free oxygen molecules will inactivate this nitrogenous enzyme irreversibly. Okay. So it's important to keep this nitrogenous from this oxygen-rich environment. The legume solution is to produce this so-called legume-globin, which contains a heme, which then binds a free oxygen molecules. The function of this legume-globin is just like our own hemoglobin, in that it binds oxygen, and in this case, by binding it, it keeps it at a low, sufficiently low level, so that it's not able to inactivate the nitrogenous enzyme. So it's possible that this is done exclusively in a nodule to sequester this nitrogen-sensitive environment from other cellular metabolism that the plant does, which either produces or consumes a lot of nitrogen. Right. So this need to protect the bacterial nitrogen enzyme also requires this bacteria to be sealed off from the ambient environment. And this in most modern legumes is accomplished by the release of bacteria from the infection thread, which is still connected to the outside environment into the cytosolic environment, and then surrounded with a host-derived membrane. So now you can see that inside is a bacteria with its nitrogenous enzyme, and outside is surrounded by cytoplasm full of hemoglobin to protect this enzyme. Okay, so this the fungal symbiosis does not do. Right? So what are the mechanisms that allow this bacteria to leave the infection thread and be engulfed, if you like, by the host cytoplasm? The short answer is we don't know, but we now have some clue to begin to address this question. So just like in that diagram, this infection thread in real life is also more or less a straightforward structure. It goes into this wild type cell and releases bacteria, which forms a symbiosome. They are alive because they have this green fluorescence, and the red fluorescence indicates the host nuclei. Okay, we found a mutant that is unable to do this. So in this magnified image, you can see that there is infection thread. It's growing and growing and growing. It just never stops. Okay? The bacteria is kept inside the infection thread until it dies. You can see that by their red fluorescence. So we call this mutant LIP for life in prison, because the bacteria never released from inside this structure. Now, knowing what gene is defective in this mutant could help us have a better understanding of the requirement and the conditions that allow the bacteria to leave the infection thread and enter the host cytoplasm. Now, whatever the function of the process, once the bacteria is inside the host cytoplasm, then it's under total control by the host. Okay? And we now know that this is accomplished by directed protein secretion from the host cell to the bacteria. Okay? So in this diagram, there are two proteins that are important. One is a signal peptidase complex, and the other one is a syntaxin. There are two here. One is called A, and one is called C. Those are two protein receptors for secretory vesicles, except that the A version localized to the symbiosome specifically, and it ensures that the vesicles packed with material destined for the bacteria are delivered to the symbiosome rather than the extracellular space. Okay. And if you knock out or disrupt either one of them, then the bacteria do not develop. We then also show that the symbiotic syntaxin, called C132A, is also required for the arbuscular mycorrhizal symbiosis, in that when you silence it using INRI, then you no longer see these very well-developed arbuscules. So that tells us that in the interaction with beneficial microbes, whether it being fungus or nitrogen-fixing bacteria, protein secretion to this host microbe interface is really, really important. Okay, and of course, what is really transported really depends on the nature of the microbe and what the host wants with it. In the case of arbuscular mycorrhizal symbiosis, the fungal structure, the arbuscule, is very short-lived. It stays 1 or 2 days before it's degraded. On the other hand, this rhizobial symbiosis, the symbiosomes are much longer lived. Not only that, they also undergo this very dramatic transformation when it's inside the host cell. Okay, so this bacteria here will grow by 1,000-fold. Its genome will amplify. Its membrane will be permeabilized. And then most important, it will no longer multiply. Okay. And we now know that this is accomplished in part by a very precise application of a set of antimicrobial peptides. Okay. So those AMPs are delivered at a dose to the bacteria, not to kill it, okay, but rather to take over its physiology so that the bacteria no longer does what it does in evolution, which is to proliferate uncontrollably, but to do what the plant requires it to do, which is to fix nitrogen continuously, nonstop. So in a sense, we can see that those bacteria are domesticated by the host, in that the host cell goes to the environment, captures a wild bacterium, put it inside its cytoplasm, raises it, and then use it for its own advantage. Okay. So in summary, to engage with nitrogen-fixing bacteria, legumes co-opted the perception system and conduit that's originally derived from arbuscular mycorrhizal fungi. They repurposed root development genes to construct this dedicated symbiotic organ called nodule, and they developed a new mechanism to internalize the bacteria, where they use directed protein secretion to control the fate of the now intracellular bacteria. We are ignorant in many aspects of this whole process, which is the reason we are kept busy working every day. So finally, I'll now acknowledge the people, both our collaborators, current members, as well as former members who contribute to the work, as well as our funding sources. I'll stop here and answer questions after all the talks are over. Thank you.